Okay, hello. In this video, I'm just going to show how you can download a genome yourself and parse it using Ruby, which is a common scripting language, pretty similar to Python. So if you know Python, you can probably watch this video. I'm going to walk through the script, show how it works. So basically, this is for educational purposes. If you're just curious to see how far you can go with a basic knowledge of programming or scripting to understand basic genomics, this is where to start. And so I'm just learning too, and please comment below or let me know if I get anything wrong here. Okay, so let's get started. Okay, so first off, I'm just going to show you a genome so this is for porcine circovirus. You can see up here the first line is basically a header showing the name of the organism. This is essentially a one long string, which is the complete DNA of the virus. And so this is basically the shortest one I could find, and I chose that just for simplicity, so it's kind of easier to manually inspect results and see what's going on. So you can see here that this genome is basically made up of one of three elements. It's A, T, G, one of four. It's A, T, G, or C. And those are generally split up into what are called triplets or codons. So I'll get to that in a second, but I just want to show you where you can get this file to start with. So let's just start at Google here, and NCBI genome database. Right up here we can search for the name of the organism we're interested in. Okay, that's what we want. Genome assembly. I believe these are different instances of the virus being sequenced. So I'm going to just pick one of them there. And right up here we can see download sequences in FASTA format for genome. Okay, so we get a file. You unzip that file. You get something that looks like this, a .fna file. And basically that's just a raw text file. So that's what we're looking at here. And the script that I'm going to write is going to take just this file as input, and it's going to try to parse this and tell us what type of proteins can or would be expressed by this. Okay, so basically we have to split this up and convert it into amino acid chains. That's our goal here. That's the entire goal of this script. Okay, so the molecular mechanism for parsing this and turning it into a protein, the first step is being kind of iterated by a ribosome. So a ribosome scans this. It's a molecule it attaches on, and it decides where to start transcribing and where to stop. So transcribing is what you call the process of actually converting this sequence into a working protein. Okay, so the ribosome is either going to start going in one direction or the other direction. So it can start at the start of it, at the head of it, or at the end of it, the tail of it down here. And that's basically because DNA is symmetrical. It's like it has a kind of reverse flipped symmetry to it. So you don't see that here in this text file. It looks like it's just one linear chain. But if you want to picture this physically, imagine this entire thing kind of flattened out into one line, and then if you copied it and pasted it below it, and then reversed it, so it was going in the opposite direction. So you would have like a mirror left-right symmetry like this, and then you do what's called the complement. So when you have a particular DNA sequence, certain characters are always across from other characters. So that's what's called a complement operation. Okay, so to summarize all of that, essentially this can be parsed from this end or from the other end, but when you're parsing it from the other end, you're not seeing exactly these characters, right? So you're not starting off seeing TCG, you're starting off seeing the complement of TCG. So I'll show you more specifically what that looks like in the script in a moment. Okay, so just to back it up, basically you have a ribosome. You're iterating along this, looking for where to start and where to stop in order to transcribe a protein. And the thing that you start and stop on, that's called an open reference frame, or an ORF. So those are basically the units. So we might have an ORF that starts there and ends there, and then this gets transcribed into a protein. Okay, so in addition to being able to start at the start or start at the tail, you can start with an offset. So this ribosome, it's iterating down in steps of 3. And that would be a 0 offset. So we're starting right at the right at the start, can't go any earlier, and every 3 we're breaking it into a codon and looking at it like that. The ribosome can also start with an offset of 1, so that's like this, or it can start with an offset of 2, so that's like this. Okay, so hopefully that's clear enough. And the same thing is true for the opposite end. So you can start at the tail with an offset of 0, 1, or 2. Okay, so overall that's 6 different ways you can parse this. And that is parsing the raw genome into codons. So you map essentially one linear string into 6 different views on that string, which are lists of 3 elements, which are called triplets or codons. So kind of the most basic one with 0 offset starting from the front. The first codon would be GTA, second codon would be GTA, third codon is TTA, and so on. Okay, so what do you do with that? Basically, there is a codon or set of codons that tends to start an open reference frame. And I say tends because that's not a 100% guaranteed relationship, and it's a little bit complex in reality which codons actually start an open reference frame. So essentially what I'm going to be doing in this script is producing candidates. So what I mean by that is I believe that the output of this script will include all of the proteins which could be produced and possibly more. It's not going to contain less, but I think in reality there are factors that mean that a lot of these things in open reference frames are not actually expressed as proteins, and I'm not going to go into all that detail here. Okay, but essentially what I was saying is there's a particular type of, there's a particular start codon or codons that tends to start an open reference frame, and there's a set that ends an open reference frame. And the ones that end an open reference frame, my understanding is those are more guaranteed. So it's a little bit looser what type of codons are going to start and to make that judgment, but it's a little bit more predictable and deterministic whether a codon is going to stop an open reference frame. Okay, so with that, let's just get right into the script here. Right here, star codons. Okay, so this is 2 triplets, and just very simple, one of these 2, either of these 2, may start an open reference frame. These are end codons, and any of these may end a open reference frame. So essentially you're looking for a list of, so once you've broken it up into triplets using one of your 6 possibilities, you're going to iterate down that, looking for every sequence that starts with one of these and ends with one of these. So this is where it gets a little bit hard to show it manually in Notepad++, because it's not easy to perform that operation of splitting into 3s, 6 ways, and blah, blah, blah. But, once you have a scripting language, it's easy enough. Okay, and then the other thing here, you can see minimum Orf length, and this, as far as I know, is just a statistical thing. This is not exactly guaranteed. I think you might have cases where you can have shorter ones, but I think the majority are longer than this. So we're just going to use this for simplification right now. Okay, what do we have here? So this is a mapping from all codons or triplets to amino acid. So I don't know the name of every amino acid off the top of my head, but essentially it's just a fairly simple dictionary, what you would call a dictionary in Python, or here it's this is a hash in Ruby. Okay, anyway, so let's just take a look here. I'll show you some of the operations I was talking about when we were looking at the genome above. Okay, so this function here, loose start heart end, is kind of one of the first interesting ones to look at, and I'll just open up a new window here to show you exactly how that's operating. So let's just take a look at the code for a second first. So we're using the inject method on self on a triplet array. So a triplet array is after this has been split up into triplets. So what I mentioned earlier, like this. Okay, so let's just kind of look at this as an example. So we have an array composed of these triplets, and we're doing an inject on that, which is kind of similar to basically like map reduce, and we're starting with an empty array. So essentially what this means is that we're starting with an empty list structure, and we're going to be kind of creating that list structure or adding stuff to it as we step down those three things. And the list structure we're creating in this case is the list of all open reference frames. So this is kind of the meaty part of actually scanning it, looking for those starts and ends. And it's a fairly short sequence here, but there's a lot of stuff going on, so it's important to understand this in detail. Okay, so for the inject operation, that gives you a pair, which is what's being accumulated. That's all of our open reference frames that we find. And this is each new triplet that we look at. So this is going to kind of add up with open reference frames, and this is what's being tacked on to each particular open reference frame. Okay, so what's going on here is, if you imagine being the ribosome stepping down, that particular line there is adding on every triplet we see to whichever open reference frames that we're currently inside. So the interesting thing there is we may be inside more than one open reference frame. So just for the sake of argument, you could have a start codon here and a start codon here. And because there's kind of this loose nature of parsing open reference frames, both of those could be valid starts to an open reference frame. So you could have a protein expressed starting here, or you could have a protein expressed starting there. It's kind of an OR relationship, or a whitelist, you could say. Whereas the stop codons are more of a blacklist type of behavior. So what I mean by that is, if you have a stop codon there and a stop codon there, you're not going to have a valid open reference frame that extends up to here. It's going to stop right there. Okay, so this line here, essentially what this is saying is, if you're inside an open reference frame already, and it's not an end codon, tack it on to all the ones that are currently open. And what this is saying is, if you're just seeing a start codon, definitely start a new open reference frame. So that's regardless of whether or not you're currently in another open reference frame. Okay, so basically that's what's going on with this function. This one is a little simpler, but still good to look at. So to protein, basically this takes a list of triplets, so like something you see up here, and converts it into the equivalent amino acid character chain. So that's essentially using what's going on up there. So let's just look at that in detail here. Taking self, which is the list of triplets, and looking up in genetic code after converting to RNA. So if you just look at this carefully here, for example, the UAU, you can see we don't actually have those characters in here. We have no U's. So that is described in an operation called transcription. So you can see here, when we go from DNA to RNA, that ribosome converts thiamine to uracil. And that's where you get the U's from in this map or hash structure up here. Okay, so where was I? Okay, so two triplets, that's fairly straightforward. That's, I think, what I described earlier, basically where you're splitting up in threes, but you can do it different ways. Okay, so this function here, this is basically mapping a single string to the 6 possible ways of it being scanned by a ribosome. So 0 offset, 1 offset, and 2 offset forwards, and 0 offset, 1 offset, and 2 offset complement backwards. And then it just returns an array of those 6 triplet lists. Okay, so basically the file, this part here is a little bit less exciting, not really theoretical, but it's just opening up that file, taking your inputs, putting it into the functions up above, and then printing it out for you. So kind of from here to the top is the actual theoretical meat of parsing the genome, and the stuff down below is just sort of front end, you could say. Okay, so I'm going to run this script here on this genome that I've downloaded, just for the sake of demonstration. You can see what the output looks like. So there it is. Just raw chains of amino acids. But this is essentially a starting point, and from there you can look at these genomes, you can search for them online, you can try to understand what they're associated with. Basically, it's the first step to functional understanding. So I hope to go a little bit further with this myself. I'm just scratching the surface. But please ask any questions and let me know if I've got anything wrong here. I'm happy to receive corrections, or if I'm misunderstanding something, please clarify that. Okay, thank you.